Welcome to part 10 of this video on the immune system. Okay, so we're currently in the process of discussing the initiation of the adaptive immune system, and we have so far discussed antigen-presenting cells and how they're going to present fragments of antigens on their surface, and we've seen that if the pathogen is intracellular, then what will happen is we can present fragments of that pathogen's antigens on MHC Class 1 complexes on the surface of an antigen-presenting cell, and these complexes are now going to initiate cell-mediated adaptive immune responses, whereas if the pathogen is extracellular, we're going to present fragments of the pathogen's antigens on the surface of antigen-presenting cells attached to MHC Class 2 complexes instead. So what I now want to do is I want to study MHC Class 1 and MHC Class 2 complexes in more detail, because there is complexities here. So firstly, what I want to do is discuss the structure of an MHC Class 1 complex and the structure of an MHC Class 2 complex. Then I want to discuss the genetics. I want to go to the MHC Locus on chromosome 6 and discuss the fact that there are actually 3 different types of MHC Class 1 and 3 different types of MHC Class 2. Then I want to talk about the fact that each of the 3 different types is extremely polymorphic. And then finally, I want to discuss what is it that determines whether a certain MHC Class 1 or Class 2 molecule can present a certain peptide fragment. What determines which peptide fragments they can actually bind to. Okay, so let's start off then with the simple bit, which is just discussing the structure of MHC Class 1 and MHC Class 2. So we'll start off again with with MHC Class 1. Okay, so Major Histocompatibility Complex Class 1. So the MHC Class 1 complex, it consists of overall 2 proteins that are bound together. So one of these proteins is known as the alpha chain, and the other protein is known as the beta-2 microglobulin. So let me just draw this now in. So let's say this is the cell membrane, and we'll imagine that we're showing an MHC Class 1 complex on the surface of our antigen-presenting cell. So let's say this is the cell membrane here. So I'm going to start by drawing the alpha chain, and the way I'm going to draw it is like so. So it has 3 major domains, which I'm going to draw as these squares. So here is the amino terminus of the peptide. Then the first important domain that we have is known as the alpha 1 domain. Then you have a second really important domain known as the alpha 2 domain. So it's very simple naming here. Remember, this polypeptide is known as the alpha chain. And then a third important domain. All of these important domains are extracellularly called the alpha 3 domain. Then you have the membrane spanning alpha helix, and then the C terminus of this alpha chain is going to be intracellularly. So this is the alpha chain that's going to make up our MHC Class 1 complex. Okay, so I'll just colour in the alpha chain here in blue. So that's the alpha 1 domain, the alpha 2 domain, and then the alpha 3 domain. And this is the big polypeptide that makes up the MHC Class 1 complex. The second polypeptide that makes up the MHC Class 1 complex is much smaller, and that has 1 equivalent domain to these 3 domains that the alpha chain has. And this is known as the beta-2 microglobulin, often just written as beta-2M, like so. So here's the N terminus and here's the C terminus, and that's not an integral membrane protein, it's just bound to the alpha chain, like so. So this is the beta-2 microglobulin here in green. So this is often just abbreviated down to beta-2 with a dash and then an M. Ideally there would be a dash in there, I've just put it in. I didn't put it in previously because it's a little bit squashed. And this stands for the beta-2 microglobulin, and it's an important part of the MHC Class 1 complexes. Okay, and it's coloured in there in green. Okay, so that's what an MHC Class 1 complex consists of. It consists of an alpha chain with these 3 important domains, alpha 1, alpha 2, and alpha 3, and then a beta-2 microglobulin that's completely extracellularly and is just bound to the alpha chain. And I'll just stress, this is the extracellular side, so this is the extracellular fluid, and this is the cytoplasmic side here. Okay, now the next question is, where is the peptide binding groove of this MHC class 1 complex? Because remember, when we previously looked at the MHC class 1 complex, the incredibly important feature of it is that it has that groove for binding peptide fragments that are between 8 and 10 amino acids in it. The peptide binding groove is actually in between the alpha 1 and alpha 2 domains. So effectively it's this little slot here. So this is where, I'll just label it up, the peptide binding groove is going to be. Peptide binding groove. And this picture in a way doesn't show this brilliantly, so what I would like to do is just draw another picture to give you a better understanding of what the peptide binding groove is actually going to look like. So let's imagine that we are now a little man, and we're going to stand up here where I've put the title peptide binding groove, and we're going to look down at the top of the MHC class 1 complex. What would we see? We'd see something along the lines of this. Okay? Here would be the peptide binding groove, and then on either side you'd have these two important domains. Let's say this is the alpha 1 domain, this is the alpha 2 domain, like so. So you've got the alpha 1 domain on this side, like so, and then the alpha 2 domain on the other side, and you can see that in between the two domains you have this little groove running, and that's the groove where this peptide fragment between 8 and 10 amino acids can actually sit, like so. So this is a peptide fragment between 8 and 10 amino acids. Okay, so that's the structure then of an MHC class 1 protein. We have an alpha chain, a beta 2 microglobulin, and the peptide binding groove is made up by the alpha 1 and alpha 2 domains of the alpha chain. Okay, so just two proteins make up an MHC complex. And it's the same for MHC class 2. MHC class 2 is just made up of two separate proteins. However, it's more equally divided in the case of MHC class 2. So let me now draw you a picture of MHC class 2, and we'll compare and contrast here. Okay, so MHC class 2 now. Right. So MHC class 2 is again going to consist of just two proteins, but as I say, it's more equally divided. You're going to have an alpha chain and a beta chain this time. So here's the cell membrane, and now I'll start by drawing the alpha chain. So here's the amino terminus of the alpha chain, and then again we're going to have important domains, which I'm just going to show as boxes here, and they're going to be called really simply alpha 1 and then alpha 2, and I should stress that this alpha chain that we're using to make this MHC class 2 protein, it's not the same protein as we had for the alpha chain of MHC class 1. This was an alpha chain of an MHC class 1 complex. This is an alpha chain of an MHC class 2 complex, a very different polypeptide. Okay, and it only has two of these important domains. It has an alpha 1 domain and an alpha 2 domain here, then its membrane spanning alpha helix, and then its C-terminus on the intracellular side. Okay, then the other protein is then the beta 2 chain, sorry, not the beta 2 chain, the beta chain this time. So here, I'll just label this one up firstly. This is the alpha chain, here in blue, with its two important domains. And then over here we're going to have the beta chain, and the beta chain is going to be just like the alpha chain. It's going to have two important domains, like so. Then a membrane spanning alpha helix, and its C-terminus on the intracellular side. So it's basically, as far as my picture is concerned, just a mirror image of the alpha chain. Of course, they are different polypeptides. They will have different sequences of amino acids and slightly different structures. So here is the beta 1 domain, and here is the beta 2 domain. But as far as these important domains are concerned, they're very similar. Okay, so this is the beta chain of the MHC class 2 protein, here in green, and the peptide binding groove is then going to be up between the alpha 1 domain and the beta 1 domain. So the peptide binding groove would be here, so you'd have a peptide bound in there, and that's just, we're just seeing the peptide along its well, we're not seeing it along its axis, we've just taken a section through it, so we're just seeing a dot, because we're just seeing, we're not seeing it in its longitudinal direction, we're just seeing a little cross section of it, so we've effectively just cut through, and we're just seeing the little dot for the peptide fragment there. Okay, right, so that is what an MHC class 2 complex looks like. So, the essential message here is that both MHC class 1 and MHC class 2 are made out of two proteins. MHC class 1 is made out of an alpha chain and a beta 2 microglobulin. MHC class 2 is made out of an alpha chain and a beta chain. And MHC class 1, the only bit that actually makes up the peptide binding groove is the alpha chain. Whereas MHC class 2, both the alpha chain and the beta chain are involved in making the peptide binding groove. So that's the structure of these. What I now want to talk about is the genetics of the major histocompatibility complex proteins, because this is where it starts to get complicated, because the reality is that there are different types of MHC class 1 protein complexes and there are different types of MHC class 2 protein complexes that are all grouped together because they do the same thing, but they are fundamentally made out of different polypeptides. And this is going to be made very obvious when I actually show you the genetics of this, and we've got multiple different genes. Okay, so let's now do the genetics then. So what I'm going to do is I'm going to go and have a look at chromosome 6. So I go to chromosome 6, and on chromosome 6 there is a specific locus known as the major histocompatibility complex locus. So on chromosome 6 I can find a little region of that chromosome which is known as the MHC locus. And of course you have two chromosome 6s. You have a paternal chromosome 6 and a maternal chromosome 6, and on both of those chromosome 6s you will have this MHC locus, and this MHC locus contains all the DNA which is going to make both MHC class 1 and MHC class 2 complex. Actually that's not quite true. The beta 2 microglobulin I don't think is in there, but it's going to contain most of the DNA that makes the major histocompatibility complexes. Okay, so let me now draw this on here. The important bits that are going to determine the peptide binding grooves are certainly going to be in this MHC locus. So this is going to represent one of my chromosome 6s, so this is the double-stranded DNA here. Okay, so I'm now going to this specific region of chromosome 6 where the MHC locus is present. And in the MHC locus I'm going to have 6 important regions that I'm going to put on here. 3 of them are going to be important in making MHC class 2 protein complexes, and 3 of them are going to be important in making MHC class 1 protein complexes. So let me now put these in. So we'll have one here, we'll have our second one here, our third one here, fourth one here, fifth one here, and sixth one here. Okay, so these first 3 here, these are going to be involved in producing MHC class 2 protein complexes. So I'll colour them all in green because they're going to be involved in producing MHC class 2 complexes. And remember, you'll have this on both chromosome 6s. So on both chromosome 6s you have an MHC locus, and you'll have these 3 regions in both of those MHC locuses. Okay, so these regions are known as the HLA-DP, HLA-DQ, and HLA-DR loci, respectively. So this is known as the DP, this is known as the DQ, and this is known as the DR. And in front of that you can put the full title HLA. HLA is another name for MHC. So it stands for human leukocyte antigen because it was found on the surface of leukocytes. So human leukocyte antigen. It's just another name for MHC. So MHC, human leukocyte antigen, they're referring to the same protein complexes. Okay, so these are the HLA-DP locus, this is the HLA-DP locus, this is the HLA-DQ locus, and this is the HLA-DR locus. Now what are these gene loci doing? And notice I'm not calling them genes, you would refer to them as loci rather than genes, and the reason is that actually each one of these regions contains multiple sections that we would refer to as genes, and I hope that will become a bit more obvious in just a moment. So the HLA-DP locus here, this has genes in it for producing both alpha chains and beta chains of MHC class 2 complexes. So you can produce from the HLA-DP locus MHC class 2 protein complexes. So HLA-DP is a type of MHC class 2. It's the same for DQ. DQ, this locus here, has regions, genes, for making alpha chains and beta chains. And if you make an MHC class 2 protein complex out of alpha chains and beta chains from this gene locus, you call that MHC class 2 molecule an HLA-DQ MHC class 2 molecule. Finally, it's the same for DR. This region here, this locus, will have genes for making both alpha chains and beta chains, and if you use alpha chains and beta chains produced from genes in this locus, you will call the MHC class 2 molecule an MHC class 2 molecule of the HLA-DR type. Okay, so overall, there are 3 different types of MHC class 2 protein complexes that you can actually produce. You can produce HLA-DP MHC class 2 complexes, you can produce HLA-DQ and HLA-DR as well. So 3 different types of MHC class 2, but they all perform the same function. They all perform the function that we've just been through. This function of presenting antigen fragments from extracellular pathogens, they all do this. So when I've drawn this yellow molecule here, it could have been any of those 3 types. It could have been HLA-DP, HLA-DQ, HLA-DR. The story would have been exactly the same. And in fact, in the rough endoplasmic reticulum, you'll produce a variety of all of these different HLA, DR, DP, DQ, MHC class 2s, and they'll all be put on the surface with antigen fragments in them. Okay, right. So, different types of MHC class 2, and now it's the same story for MHC class 1, so we have 3 loci then for making MHC class 1 complexes as well. So this one is known as HLA-B, this one is known then as HLA-C, and this one is known as HLA-A. So you've got HLA-A, HLA-B, HLA-C, and then all of the HLA-Ds, which are the MHC class 2s. So I'll just colour these ones in here. So here we have HLA-B locus, here we have the HLA-C locus, and this is the order that they appear in, and here we have, or the order that they've been named in, and this is the HLA-A locus, so BCA. Okay, and each of these loci will produce, well, will have genes in it for making alpha chains. So if you, but this is the important thing, they don't have the gene for making the beta-2 microglobulin. The gene for the beta-2 microglobulin is elsewhere. I don't know specifically where the beta-2 microglobulin gene actually is, but it's elsewhere, and you just have 1 gene for beta-2 microglobulin. Okay, you'll of course have 2 copies of it. You'll have one on a paternal chromosome and one on a maternal chromosome. Sorry, one on a paternal and one on a maternal chromosome. But you just in, one complete set of chromosomes, so from 1 to 22, you won't have multiple copies of beta-2 microglobulin like you do have multiple copies of genes for making MHC class 2 complexes and also the alpha chains of MHC class 1 complexes. So the beta-2 microglobulin gene is elsewhere, and it's just producing beta-2 microglobulin proteins. These regions here, these produce the alpha chains. So you have the HLA-B locus, which will produce alpha chains. You have the HLA-C locus, which will produce alpha chains. And you have the HLA-A locus, which will produce, again, different alpha chains. So you can produce 3 different types of MHC class 1. You can produce HLA-B type MHC class 1, which is where you've used the alpha chain from the HLA-B locus in the MHC locus here. You can produce HLA-C type MHC class 1 protein complexes, which is where you've used alpha chains from the HLA-C locus here. And you can produce HLA-A type MHC class 1 protein complexes, which is where you've used alpha chains from the HLA-A locus of the MHC locus here. Okay, right. So that's the first big concept that I want you to understand, that there are different types of MHC class 1 protein complexes and MHC class 2 protein complexes, but that all these different types perform the same function of presenting antigen fragments from intracellular pathogens in the case of MHC class 1 protein complexes, and from extracellular pathogens in the case of MHC class 2 protein complexes. Okay, right. And remember also that the MHC class 1 complexes, they will like to present antigen fragments between 8 and 10 amino acids, whereas the MHC class 2 protein complexes, they will like to present peptide fragments between 12 and 20 amino acids. Okay, so that's the first difficult concept. Next difficult concept is that all of these loci here, these 6 loci of the MHC locus, they are all extremely polymorphic. So what does this word mean? Poly means many, morphic means form. So they have lots of different forms. In the context of genetics, polymorphic, with reference to a certain locus of DNA, means that if you were to go into, if you were to sample a huge population of humans, let's say we took a sample of 1,000 humans and we were to sequence these loci in all of those 1,000 humans, you would find huge variation. It would be very different in different people. There are a huge number of different alleles of all these regions. So remember, alleles are the variants of loci of DNA. Okay, so there are a huge number of different alleles for the HLA-DP locus. There are a huge number of different alleles for the HLA-DQ locus. There are a huge number of different alleles for the HLA-DR locus, and the same for all of these MHC class 1 ones as well. A huge number of different alleles for HLA-B loci, a huge number of different alleles for HLA-C loci, and a huge number of different alleles for HLA-A loci. Okay, so this makes it even more complicated. The beta-2 microglobulin gene, as well as there just being one copy of it, the simple thing is that it's not polymorphic. Everyone would have the same identical, pretty much, gene for beta-2 microglobulin. We all produce identical beta-2 microglobulin proteins, whereas we don't produce identical alpha chains for MHC class 1 or alpha and beta chains for MHC class 2, because there are loads of different alleles for these. Okay, so firstly let me just give you the way that different alleles are named up. So, for the HLA locus, HLA-A locus then, so we're now talking about the HLA-A locus, and this is the way it's written. You put HLA and then you put a dash, and then you either put A, B, C, DP, DQ, DR, whichever one you're talking about. Because there are loads of different alleles here, the way it's named is that you put a great big number after this. So if you're reading research papers on some sort of autoimmune disease or any sort of disease which is hugely driven by the immune system, you will not be able to avoid seeing things like this, where there are certain genes that have been found to be associated with the disease or associated with better prognosis in the disease, and you will see things like this. You will see HLA and either A, B, C, DP, DQ, DR, and then a great big mess after here. A great big number, but unfortunately it's not just a number. They put colons and stars, so you'll see a great big mess. So if you're lucky, you might see something like this, HLA-A1, and that's just, the number is telling you which, it's referring to a specific allele of HLA-A. If you're unlucky, you'll see some massive great mess here, like 1 star, 26 colon 21, and I'm just making this up, but you'll see something horrendous. What I want you to take away from this is that all this great big mess after the HLA, whatever letter A, B, C, DP, DQ, DR bit means, is it tells you which specific allele you are dealing with. So this is the name of the specific allele of that locus. Okay, and you will see this commonly if you read the literature. Okay, so that's the naming of the different alleles. So hopefully you'll now understand the fact that we have lots of different genes coding for MHC class 1 and MHC class 2 proteins, and that in these little loci which code for MHC class 1 and MHC class 2 protein complexes, there are loads of different alleles for them, and therefore we're all producing slightly different MHC class 1 and MHC class 2 protein complexes. Now, why is this important? Well, it becomes important when we start considering what is the specificity for binding a peptide fragment. What determines whether a certain MHC class 1 protein complex or a certain MHC class 2 complex can bind a certain peptide fragment in its peptide binding groove? And that's the topic that I want to discuss now. So let's say that we have a certain type of HLA, a certain type of MHC complex. So just to take a specific example, let's say that I have an HLA-A MHC class 1 complex, and let's say it's an HLA-A1. Okay, so let's say I have used this specific allele of the HLA-A locus, which is the HLA-A1 allele, so that's a specific version of this locus here, to make my alpha chain. I've then used the beta-2 microglobulin gene to make a beta-2 microglobulin protein. Remember, beta-2 microglobulin isn't polymorphic, so it's the same in all humans, and the gene is always pretty much identical. Okay, so I've now got some MHC class 1 here. And I'll just draw this simply. So I'll go back to drawing a simpler picture of MHC class 1. So rather than draw the separate alpha and beta-2 microglobulin, I'll just draw it like so. So here we go. This is going to represent my MHC class 1, and I'll go back to representing it in pink as I did previously, I think. Okay, so here it is. So it's been built from an alpha chain made from this specific allele of the HLA-A locus, and then it's got a beta-2 microglobulin, but that's not polymorphic, so that's always the same. Okay, what determines which specific peptides this can bind? Well, we know it will only bind peptide fragments between 8 and 10 amino acids. But can it bind any old peptide fragment between 8 and 10 amino acids? The answer is no. So which peptide fragments can it actually bind? Well, the answer is that the peptide fragment has to have certain amino acids at certain positions to be able to bind in the peptide binding fragment of this specific MHC class 1 complex. And the specific amino acids that you'll need at specific positions will change depending on which specific MHC complex you're dealing with. Okay, so let me explain this in a little bit more detail. So this is the concept that is referred to as the concept of anchor residues are needed in the peptide fragment. So the idea is this: on the surface of the peptide binding groove, there are certain little pockets that that certain amino acid residues have to stick into in order for the peptide fragment to be able to bind here. So in order for the peptide fragment to be able to sit in this peptide binding groove here, it has to have certain amino acids at certain positions which can fit into these peptide amino acid binding pockets. So these are the concept of binding pockets. And these residues that you have to have in certain positions, these are called the anchor residues. So let's just say that we're going to bind a peptide of length 8 amino acids in here. So let's just divide this up into 8. So 1, 2, 3, 4, 5, 6, 7, 8. Okay, so here are the 8 amino acids. So this is one amino acid, this is another, third, fourth, fifth, sixth, seventh, eighth. And this is now sitting in our peptide binding groove here. I'll just colour this in, so we'll have our peptide binding groove here. Okay, and here's our little peptide here. So potentially, in order for this peptide to bind here, and I'm just going to make this up, I don't know what the specific anchor residues needed for HLA-A1 are, but I'm just going to make it up to get the concept across. So in order for this peptide to bind in this peptide binding groove of MHC class 1 of this specific type, maybe it needs to have, let's say, a glutamate amino acid residue here to bind in some specific pocket that is here on the peptide binding groove. So maybe there's some specific little pocket here that the glutamate's R group needs to fit in here in order for you to be able to bind. And maybe you also need to have an arginine at this position. So what position is that? That's position 5. So a glutamate at position 2 and an arginine at position 5. And this arginine might have to bind in some specific little binding pocket here on the peptide binding groove as well. So here's a little pocket and here's a little pocket. So these are the specificities. This is what determines when a peptide fragment can actually bind in this binding groove. There are certain little binding pockets on the surface of the peptide binding groove, and in order for the peptide to be able to bind, it doesn't matter what's in position 1 or position 3 or position 4 or position 6 or position 7 or position 8. You can have whatever you like there, just as long as in position 2 and position 5 you have these specific amino acid residues, a glutamate and an arginine, which can fit into these peptide binding pockets. Then the peptide fragment will bind. And these residues, the glutamate at position 2 and the arginine at position 5, these are called the anchor residues because they anchor the peptide fragment in the peptide binding groove. Okay, so each of these different MHC class 1 and MHC class 2 proteins that you can imagine producing from the different types of MHC class 1 and the different types of MHC class 2 and all of the different alleles of each of those different types will have different requirements of anchor residues. They'll require different anchor residues because they'll have slightly different peptide binding grooves, and therefore they'll have different binding pockets that need to have the anchor residues fitting in. So the requirement for a fragment to bind in to the peptide binding groove is different for these different types of MHC class 1 and MHC class 2. So why does this matter? Well, this is the way that we initiate the adaptive immune response. How many different MHC class 1 and MHC class 2s will you be able to produce? Well, think about this. We have these 3 different loci for MHC class 2 and 3 different loci for MHC class 1. We also, remember, have the other chromosome 6, and we'll have the identical thing on that other chromosome 6. And of course the alleles for each of these that we have doesn't need to be identical on the paternal and maternal chromosomes. So in total, we could produce then 1, 2, 3, 4, 5, 6, I'm imagining we've got the other chromosome here. We could produce 6 different forms of MHC class 2 with different specificities, i.e. they require different anchor residues to bind the peptide fragment. And the same for MHC class 1, 6 different types of MHC class 1 that we could produce. Okay, so we're only going to be able to then show peptide fragments with the specificities of those different MHC class 1s and MHC class 2s. And this is the way that we initiate the adaptive immune response. So it must have a huge bearing, surely, because it's going to affect our ability to launch adaptive immune responses against certain pathogens. Because pathogens' antigens will have peptides that either can or cannot potentially have fragments that can be presented on these MHC class 1 and MHC class 2s that we have. So in fact, our specific combination of HLA alleles is actually going to determine our ability to fight certain pathogens and also our ability to present self-antigen fragments. Because what you have to realize is that there's nothing here that stops these MHC class 1 and MHC class 2 protein complexes from presenting fragments of self-peptides. Self-peptides will occasionally have the correct combination of anchor residues to be presented on MHC class 1 and MHC class 2, and occasionally they will end up being presented on these MHC class 1 and MHC class 2 residues. It is not with the MHC molecules that the role of preventing autoimmune diseases lies. We will see mechanisms for preventing autoimmune disease later on. That's at the level of the T cells and the B cells. So MHC class 1 and MHC class 2 molecules, they can and do present fragments from self-peptides to lymphocytes, to the adaptive immune system all the time. The mechanisms by which we prevent autoimmune diseases actually coming about are by stopping ourselves from producing T and B lymphocytes that are actually capable of then responding to these self-peptide fragments that are being presented to the adaptive immune system. Okay, right, so that is all I want to say about MHC class 1 and MHC class 2. I do just want to summarise it because it is complicated. So, summarising what we have learnt here. We have learned that MHC class 1 and MHC class 2 have these structures here. MHC class 1 and MHC class 2 both consist of 2 peptides joined together. So in MHC class 1's case we have an alpha chain which is polymorphic, so there are loads of different forms for the alpha chain, and then a beta 2 microglobulin which is not polymorphic, so that will always be the same. No matter what MHC class 1 complex you go to, no matter which person you go to, this peptide is pretty much always identical. Okay, and the peptide binding groove for a peptide of between 8 and 10 amino acids is between the alpha 1 and alpha 2 domains of the alpha chain of the MHC class 1 complex. MHC class 2 protein complexes look like this. They consist of an alpha chain and a beta chain. It's more even here. Both of these are extremely polymorphic and will have different forms in different MHC class 2 molecules in the same person, and also certainly different forms between different people. And the peptide binding fragment, the peptide binding groove for the peptide fragment is between the alpha 1 and beta 1 domains of the alpha chain and beta chain, as shown here. Now, the genetics is complicated. It would be really simple if we just had one gene for an alpha chain for MHC class 1, and one gene for an alpha chain for MHC class 2, and one gene for the beta chain of MHC class 2. That would be really simple. That's not the way it works. Instead, on chromosome 6s, on both of the two chromosomes, chromosome 6s, what we have is a locus that is known as the MHC locus, the major histocompatibility complex locus. And in this locus we have 6 important different regions. 3 different regions which are important in making MHC class 2 protein complexes, and 3 different regions that are important in making MHC class 1 protein complexes. The loci important in making MHC class 2 protein complexes are known as HLA-DP, HLA-DQ, and HLA-DR. The loci important in making MHC class 1 protein complexes are known as HLA-B, HLA-C, HLA-A, as shown here. Now, in the case of these loci for MHC class 2, they contain genes for making both alpha chains and beta chains. So you can make a full MHC class 2 protein complex just from the HLA-DP locus, or just from the HLA-DQ locus, or just from the HLA-DR locus. If you do it from the HLA-DP locus, you get an HLA-DP MHC class 2, etc. for the other ones as well. Okay, so there are these 3 different types of MHC class 2 proteins then, protein complexes that you can actually produce: HLA-DP, HLA-DQ, and HLA-DR. Same goes for MHC class 1, although it's slightly more complicated here because these just have genes for the alpha chain. The beta-2 microglobulin gene is elsewhere, and remember, is not polymorphic. So if you use HLA-B to make the alpha chain, then you will end up with an HLA-B MHC class 1 protein complex. If you use HLA-C's alpha chain, you will end up with an HLA-C MHC class 1 complex, and the same for HLA-A there. So, there are different types of MHC class 1 and MHC class 2. The further complication then comes from the understanding that these gene loci are extremely polymorphic. So there are loads of different alleles for all of these 6 different loci in the human population. We've gone through the naming here, so you put the HLA and then whichever one you're talking about, whichever locus you're talking about, and then you'll see a huge, great complicated code here, which will often contain stars and colons. If you're lucky, it will just be a number like 1 or 11 or 21, but usually you end up with something that looks like this. And this is just the name for a specific allele of that locus. Okay, so don't fear these things when you see it. It's just a silly name for one of the alleles of that locus. Now, why does this become important? Well, of course, it becomes important when we consider what determines whether a certain MHC complex can present a certain peptide fragment. So if you take a specific MHC complex, so in this case we took an MHC complex of the class 1 type, which had its alpha chain produced by this specific allele, and I was just picking a random example, a nice simple example. I've got a nice simple allele name here, HLA-A1 here. This is a name for a specific allele of the HLA-A locus. This will produce us an alpha chain. If we use that alpha chain with the beta-2 microglobulin protein, we get an MHC class 1 complex. This will have, it will be picky. It will bind peptide fragments of a certain specification, and the specification is that you have to have certain anchor residues to bind to certain binding pockets on the surface of the peptide binding groove, and then it doesn't matter what residues you've got in the other positions. So we will all have in all of our cells in our body, and certainly in our antigen-presenting cells, certain MHC class 1 molecules and certain MHC class 2 molecules. And I've been through how many different types you can potentially have. You have to remember that you have 2 chromosome 6s, so you have 2 copies of this, and on the 2 different copies you could have different alleles. So overall you can produce a lot of different types of MHC class 2 protein complexes and a lot of different types of MHC class 1 protein complexes, but remember that each of those different types will have a specificity like this. I will only be able to present peptide fragments of a certain specificity to the adaptive immune system. Now, this is important because it determines our ability to fight certain pathogens, and it also determines our susceptibility to certain autoimmune diseases, our ability to present certain peptide fragments of self-peptides to the adaptive immune system. Now, as I have said, MHC proteins will all the time be presenting self-peptide fragments to the adaptive immune system. The role of preventing autoimmune disease does not lie with MHC, it lies with T cells and B cells, and we will see the mechanisms to prevent you producing T cells and B cells that can respond to self-peptide fragments. However, sometimes those mechanisms go wrong, and then importantly, it then comes down to which MHC molecules you have, because if you have managed to produce T cells or B cells that will respond to a certain self-peptide fragment, then it doesn't matter as long as the MHC complexes that you have can't present that certain peptide fragment to your T cells and B cells. So these proteins, the specific combination of alleles for MHC1 and MHC2, the specific combination of MHC1 and MHC2 molecules you have and their specificities does determine and is really important in your susceptibility to suffering from autoimmune diseases. So it is something that everyone's very interested in who is studying any kind of autoimmune disease. Okay, right. So we will have a break here, and in the next video what we will actually see now is these peptide fragments of the antigens of this pathogen that we now have on the surface of our antigen presenting cell. How are they going to activate the adaptive immune system? And for that we will have to discuss T lymphocytes. So in the next video we will start talking about T lymphocytes.